My presentation is on the effect of phospholipid liposomes on prion peptide 106 to 128 amyloidogenesis. These are the outline of my talk. I will talk about the prion disease and how prion protein are responsible for the disease. The main goal of my research is to investigate the effect of different factors, such as the concentrations and pH, on the aggregation properties of prion fragment, and to investigate the effect of phospholipid liposomes on the prion fragment 106 to 128 aggregation. Prion disease is a progressive, infectious, transmissible, fatal, and so far incurable disease of the central nervous system and is commonly known as the transmissible spongiform encephalopathies because prion-infected brain slowly becomes spongy over the time. And these are the different forms of the disease, and it is estimated that the most patient dies within 6 to 12 months of the clinical onset. Prion protein is a small membrane-bound protein which is predominantly expressed in the central nervous system. Prion protein appears to have these two isoforms, and any abnormal folding of normal prion protein leads to the formation of abnormal or pathogenic infectious form of the prion called the scrappy prion protein. And so produced scrappy prion accumulates within the brain as amyloid plaques. And these scrappy prion are highly stable and they have a very high propensity to aggregate. Also, they are enriched in the beta sheet content and are neurotoxic and infectious. In my study, prion fragment 106 to 128 is proposed as a model to investigate the fibrillar activity of the misfolded and aggregated prion. So the fragment is a very small part of the N-terminal domain of the prion protein, which is composed of 23 amino acids and is characterized by the two important domains called the N-terminal domain, which is polar head containing positively charged domain, and the other is C-terminal domain containing hydrophobic tail, especially it has the hydrophobic domain. And the intermolecular interactions, such as the ionic interactions, hydrophobic interactions, are essential for the formation and the stabilization of the amyloid fibrils. And these are the in vitro methods of the study. The peptide fragment 106 to 128 were synthesized by using the solid phase peptide synthesizer using the FMOC strategy. So synthesized crude peptide was purified using the reverse phase HPLC. And these are the chromatogram showing the peptide at a retention time of 19.9 minutes. So purified peptide, and then the molecular mass of the synthesized peptide was verified by using the Maldi-Toff mass spectroscopy. Here is a quick background on the thioflavin T binding assay. Thioflavin T is a benzothiazole dye, and it exhibits enhanced fluorescence intensity upon binding to the amyloid fibrils. And the next one is the AFM, atomic force microscopy, is a very high resolution type of scanning probe microscopy that generates the images. So these two assays, atomic force microscopy and TST binding assay, has been used to investigate the property of the prion fragment. And this work was done in order to get insight into how the different concentration of the peptide affect the aggregation process. If we see the result, The THT fluorescence intensity is maximum at 120 micromolar concentration and is negligible, almost flat at 25 macromolar concentration, suggesting that the aggregation kinetics increases with the increase in the concentration. And the AFM imaging are also inconsistent with the kinetics data. So here we can see a bunch of fibrils at 120 micromolar concentration. Here we tested the effect of different pH. If we see the result, then the aggregation process is very fast and high under basic and neutral conditions. However, the final saturation intensity gets lower at basic conditions due to the precipitation of the peptide. Interestingly, acidic medium dramatically retards the aggregation of the peptide. And if we see the AFM imaging under the pH 7.4 showed the highly dense fibrils. So the main highlight here is that the aggregation propensity is very high at pH 7 and pH 8, and this property may also rely on its single histidine, which is at position 111. So the protonated histidine in acidic conditions generates the repulsive interactions that could prevent the self-association of the peptide. And here is the study of the effect of phospholipid liposomes on the prion fragment. So this is my second goal. As I already mentioned that the prion protein is the membrane-bound protein. So here we wanted to see how the peptide interacts with the cell membrane. So most proteins are exposed to the biological membranes, and binding of peptides to the cell membranes may catalyze or inhibit the conformational changes. So liposomes are the small vesicles where the hydrophilic head groups are exposed to the uh, aqueous solvent, and the hydrophobic tails are sequestered inward to the bilayer. And the liposomes have shown as a very huge promise as a vehicle for the drug delivery system. And here we use the POPC and POPZ uh, for mimicking the cell membrane in the absence or in the presence of the cholesterol. So if we see the POPC, the head group of the POPC contains the positively charged aminocations, whereas the head group of the POPZ contains glycerol along with the phosphate group and two long hydrocarbon chains. And the next is the cholesterol. Cholesterol is a neutral lipid molecules. So if we see the phospholipid bilayer here on the right picture, topmost right picture, so the phospholipid bilayer in the absence of cholesterol is loosely packed, whereas, and also the hydrophobic core is exposed. Whereas the phospholipid bilayer in the presence of cholesterol are more condensed and are more ordered, due to which the hydrophobic core is less exposed. Coming to this slide, we prepare the 4 different liposomes using the membrane pore size 100 nanometer, and their size was verified using the dynamic light scattering. So here are the results of the interaction of the POPC in the absence or in the presence of the cholesterol. If we see the result here, then higher concentration of pure POPC moderately inhibits the aggregation process. And AFM imaging under 100 micromolar concentration showed small spherical aggregates if we compared to the control. And POPC in the presence of cholesterol enhanced the aggregation process. AFM imaging at 100 micromolar concentration showed a bunch of fibrils. So the main highlight here is that PUPC induces unfavorable repulsive interactions between the positively charged PUPC and the positively charged residues in the peptide sequence. So here are the results of interaction of POPZ in the absence and in the presence of the cholesterol. So if we see the result, then pure POPZ and the POPZ in the presence of cholesterol dramatically accelerates the aggregation of the peptide if we compared to the control. And the AFM imaging at 100 micromolar concentration showed a very dense fibrils. So the main highlight here is that a strong electrostatic interaction between the negatively charged PUPZ and the positively charged residues on the peptide occurs strongly. So this is a lipid model of POPC and POPZ without cholesterol. In case of POPC, because of the strong repulsive interaction between the positively charged peptide and the positively charged vesicles, the peptide has a very weak binding to the lipid bilayer, due to which peptide starts to self-associate with each other. However, if the hydrophobic part of the peptide interacts with the hydrophobic core of the bilayer, that could lowers the peptide stacking. Whereas in case of POPZ, lipid peptide binding is initiated by a strong electrostatic interaction, which could enhance peptide stacking one over the other rapidly. And cholesterol fits most of its part into the lipid bilayer, due to which the hydrophobic region is less exposed and do not let peptides to anchor into the lipid bilayer. Hence, cholesterol could promote the aggregation of peptides at the membrane surface. And here is a very quick summary on the concentration and binding sites. At a high concentration of PUPC or the PUPZ, a large number of binding sites are available, due to which each monomeric peptide could be able to bind to a separate vesicles that could limit peptide stacking, whereas the lower concentration could favor peptide stacking, fibrillation, and the aggregation. So now here is the conclusion. So no significant binding is observed for the Zwitter ionic membranes of the POPC, nor their mixtures with the cholesterol at its low concentration. And the pure POPC slightly slow down the aggregation at a high POPC concentration. And if we talk about the PUPZ, pure PUPZ and PUPZ with cholesterol showed fast kinetics of fibrillation. Hence, aggregation appears not to be regulated by the cholesterol-rich portion of the membrane for the PUPZ. Thank you.